associate professor here in EEB. Our speaker today is Liliana Dávalos, who is an evolutionary biologist and conservation biologist with broad interest in understanding processes and mechanisms that affect biodiversity. I met Liliana a few years ago where we both were part of a dissertation committee from a student in Stony Brook. And besides loving her first name, I also love the amazing research she and her group are doing. Liliana did her bachelor's degree in biology with emphasis in genetics at the Universidad del Valle in Colombia. She then moved to the US to attend the University of Columbia, where she obtained her MA, MPhil, and PhD degrees in ecology and evolutionary biology, as well as a certificate in environmental policy studies. She was a postdoctoral research associate at the University of Arizona and a postdoctoral fellow at the American Museum of Natural History, where she is currently also a research associate. And then Liliana joined Stony Brook University in 2008, and since 2018, she's a full professor of the Department of Ecology and Evolution and the Consortium of Interdisciplinary Environmental Research. Her research group uses phylogenetics and comparative analysis, which include genetics, genomics, transcriptomics of non-model vertebrates, to answer questions about diversification in deep time and molecular evolution of diverse traits. She also studies how conflict and governmental policies impact deforestation in South America. So it's really broad, different areas at different levels of biology and conservation. Liliana has received funding for her research from the Human Frontiers of Science program and several grants from the National Science Foundation, including a research collaborative grant to establish a network among 14 research groups to use bats as models for studying diversification and sustainability, which I really find it really exciting and interesting. Today, Liliana will share with us some of her research on molecular adaptations that underlie ecologic diversity in bats. So without further ado, I just want to thank Liliana for accepting our invitation. And Liliana, welcome to Michigan, even if it's virtually.
In distance. Thank you so much, Liliana. Thank you for inviting me, for having me. So today's talk is about sensing in the dark, which is what bats do. And there's a molecular ecology of bat senses with an asterisk, because as you're going to see, it's not just molecular ecology. And before I get started, I want to thank the National Science Foundation that has funded all of the work that you're about to see. And you see this little logo with the bat on it. That's something for a 6-year Dimensions of Biodiversity project that we had that helped us fund most of the work that you're about to see. So as Liliana explained, my lab has this very this focus that's all about biodiversity. At the very center of everything that we do is biological diversity. But we have lines of work that operate at different scales and they ask different questions. So in the blue, for example, we're looking at processes, mostly evolutionary processes that tend to increase biological diversity. And when I'm saying biological diversity, I'm not thinking just about the number of species, but also traits that become more diverse. And then on the other hand, and closer to the present and closer to sort of social and economic factors, we've got processes that tend to decrease biodiversity through the process of extinction, and in particular landscape changes. And today, as I'm presenting here, of course, it's still by far the fact that a lot of threatened species are threatened by habitat conversion. And of course, climate change is quickly rising, right? And the synergies between this climate change and habitat conversion are all coming to bear right now. So I can summarize these different lines of research along in three different big broad areas. So in the blue, how does biodiversity change in time and space, which is sort of the way my work started a long time ago when I was a graduate student. Then in the green at the center is what are these biological processes to fuel biodiversity? And I'm thinking more about genomics, and we see that little bat that's part of the Bat1K logo, which is the vertebrate genome project that seeks to sequence the genomes of all the bats that are alive right now. And then in the red is what are the social factors of environmental degradation? What is it that's driving, like why, if it seems like we have this disconnect where everyone thinks it would be really good to have more sustainability, then why doesn't it happen? Why? However, I can't really hope to have a seminar that's a reasonable length and cover all of this. So I'm just going to focus on one today, and it's what biological processes fuel biodiversity. And we're going to see, at least some of the patterns that have to do with this. As I mentioned early, this is part of a collaborative grant that we had for a number of years. And being collaborative means that it's a team sport, right? Like there's other PIs that were involved, and I'm showing them here. Betsy Dumont, who is at the University of California, Karen Sears, who's at a different University of California, and Stephen Rossiter, with whom we collaborated closely. I'm showing their picture because I think, especially if you are starting out your career, it's very important that to understand that nowadays, the way our science is being done, it really requires multiple disciplines and multiple expertise, and bringing a team together like that. All right, so some of you may not be familiar with the diversity of bats, but I wanted to show just this phenotypic diversity that we see across bats that immediately jumps out. One of the things that jumps out is that we have bats like number 4 that have giant ears, right? Or number 17, and there are others that have kind of smaller ears, right? And there are bats like number 20 that you see there that actually the eye is kind of inside the ear. The ear is kind of like taking over the rest of the face, and the eye is inside of them. And you also immediately can see that the eyes, all bats have functional eyes, contrary to popular myths. Sometimes you have giant eyes in these bats, like on number 9 or number 30. And sometimes you have really tiny, tiny eyes, like the eye that's in number 20, like sitting in the middle of that ear, like with the wrinkly of the ear. So we immediately see that there's this huge diversity, at least externally. We can see there's this huge diversity, and it's extraordinary, right? Because most of us, bats being nocturnal animals, most people who don't work on bats are not familiar with the fact that different bat species just diverge so widely. Today, I'm going to talk about this group of bats that's highlighted here in the white. And this group of bats is really interesting because, so number 2 is the fishing bat. It's one of 2 bats that have evolved the ability to catch fish and feed almost exclusively on fish. So it's a single clade that's a superfamily. The majority of the species in this superfamily correspond to leaf-nosed bats of the Americas, or neotropical leaf-nosed bats. And it includes everything from the vampire bat that you're seeing, number 3, that's a vampire bat. That's the only vertebrate that is a dedicated blood-feeding animal. If we think about blood-feeding animals, vertebrates don't really come to the top except for the vampire bats. So that's a pretty specialized diet that has evolved there. It also includes bats that have this elongated rostrum that have co-evolved with flowers to actually visit the flowers and behave like hummingbirds, but at night. And then it includes bats like number 6, which has evolved a really blunt face, and apparently has, thanks to sexual selection, there's also like this face that looks like it crashed and something melted. So this huge diversity of diets, huge diversity in the ears, huge diversity in their appearance. It's part of a single clade that we have been studying for a number of years. So a bunch of years ago, almost a decade ago, we were just kind of at the very beginning trying to think about, okay, how are they related to one another, right? And we came up with this. We were, as you can see, the elongated rostra, right? You have this nectar feeding bats. At the very bottom in green, you have all these bats that have very blunt faces, but then you also have the vampire bat that's highlighted in red, and other bats that are like the vast majority of bats, and especially the bats that we have in a place like Michigan or a place like Long Island that are insectivorous, so almost exclusively insectivorous. So these form a clade, and it's a really fascinating clade because this whole diversity has evolved in relatively little time as far as these mammals go. So today I'm going to focus on 3 senses. I'm going to focus first on the sense of vision. Then I'm going to be talking a little bit about hearing. And then I'm going to talk about my favorite sense, the sense of olfaction, which is really important and has become important in fresh new ways now that coronavirus has reshaped our thinking about our sense of smell for many people, right? So in the first case with vision, I'm going to ask a question, well, does feeding ecology shape the evolution of genes that are associated with vision, right? In terms of hearing, does the bat head impose certain trade-offs? Because a bat, our head, if you think about our heads, there's all these sensory systems that have to occupy a limited space. This real estate is limited. So does that lead to some kind of trade-off, impose some trade-off? And finally, in the sensible function, does the diversity, the molecular diversity that we see in olfactory receptor genes, is it correlated to diversity in foods? So we'll start with the first. And all of my work is grounded very much on field collections. And here's an example. So pretty much everything that you're going to see is actually based out of very elaborate work in the field that involves in the field, transporting a liquid nitrogen tank, taking it to the field, taking the samples, and actually being able, taking vouchers, taking the samples, and then being able to dissect them and keep them cold enough so that we can have RNA of very high quality and reduce the degradation. So this has taken years of traveling with a liquid nitrogen shipper, which is always always a challenge. And thanks to that, we've been able to kind of sample a lot of the diversity that I presented earlier, and a lot of the different associations that these bats have with plants or with the different prey that they have in their diet, right? So we start with an obvious hypothesis. This is Kalina Davis. She is a postdoctoral researcher, and she spearheaded this. She spearheaded this part of the project, right? So we have this obvious hypothesis. If you look at what's highlighted in red, right? It stands out that you have this diversity of diets where you have fruit, different varying quantities. As it gets darker, it means that there's more and more of that particular food item that's being eaten by this species that are at the bottom, whose phylogeny is shown at the bottom. So frugivory has evolved across these bats, this clade of bats. And as you can see, right, there's some that are kind of dedicated frugivores and others that actually mix the frugivory with other diet items like nectar. So you can see nectarivory once more. It's actually evolved a couple of times. There are two distinct branches that include this nectarivorous bats. Piscivory has evolved only once. Vertebrate carnivory has evolved a number of times as well, independently. The inclusion of this vertebrates, blood has evolved only once. And of course, there is some amount of insects in the diet for the majority, but you can see that also all of this early diverging branches are dedicated insectivores, right? Now, Phylostomidae is the family, this neotropical leaf nose bats is the family that, of course, has the most species diversity and has the most bacterial diversity. But I want to draw your attention, it's not the only game in town. There's also more moppet bats, and these are funnel-eared bats that are interesting for other reasons, for the way that they conduct their echolocation. And then there's the ancestor of the neotropical noctilionoids. Noctilionoid is the name of the superfamily. And of course, we have some other outgroups that are not being presented, but that ancestor, just to keep track. So what are the results? So we went out into the field, we collected all these samples, we actually extracted RNA from the retina of different bats, and from this retinas, and we have this comparative data set across comparative data set of genes that are being expressed in the eye, a substantial chunk of which have been associated in model organisms with vision, right? So we were thinking, well, what's going to happen is that the feeding ecology, things like the evolution of plant visiting, which is shown in green, or the evolution of this diet of figs, or the evolution of nectar feeding, those moments, right, those switches in diet are going to be correlated to a huge amount of positive selection. We're going to find signals in those nodes. We're going to find signals of positive selection that indicate that they evolved, their sensory systems were evolving at that time. So I'm going to slow down here because this is a very elaborate version of a Venn diagram, right? It's an upset plot, right? And so the first thing that stands out, or for most of us in ecology and evolutionary biology, we have phylogeny, and that phylogeny shows the different nodes that are being shown at the bottom, right? But what's interesting is that we've got this bar chart at the top that shows us the number of positively selected genes. So genes that are showing a pattern that is consistent with positive selection at these different nodes, right? And so when you see just a dot, it means that it corresponds to the number of genes that are shown only in that one group. But when you see a line connecting two dots, what's happening is that there are two groups and they overlap. So we thought, well, of course, nectar feeding is such a special thing, right? We might find evidence of parallelism, whereby a number of genes will be shared across the two, right? Seeing flowers in the dark is a very unusual thing to be doing. And there is behavioral evidence that that's what the bats are doing. But instead, what we find is highlighted there in the yellow is that we only have 4 genes that are positively selected and that are found common to the two nectar feeding groups. Now at the bottom, in the smaller bar chart, is the frequency count of the positively selected genes across that one group. So for example, for group E, which is the evolution of plant visiting, right? All of the genes that we predicted that plant visiting, right? We predicted that there would be lots and lots of genes. We actually find very few. And instead, what we find, the number of positively selected genes is the highest at nodes A and C. The neotropical noctilinoid ancestor has more than 400 positively selected genes using this method. And the next node that's most interesting is the Phylogenetic, meaning the ancestor of the neotropical leaf-nosed bats. So what we were seeing was completely contrary to what we had predicted. We had predicted that, we would be finding all of this color at the bottom, right? We would find the largest number of genes were going to be positively selected in these changes in diet, and that's not what we found. So we had to start making sense of this. And in order to make sense of this, we have to learn a little bit more about bats. The bats that we have here on Long Island and also in Michigan, they all did this. Most bats are aerial insectivores. And so you see what this bat is doing. This bat has skin that is between the legs. It's called a patagium, and it's using that patagium. It's going towards the moth. It's using that patagium to sweep the moth up and bring it to its face and eating the moth. That's what most bat species do. They are actual aerial insectivores. They go on the fly, and that's how they catch their prey. But not all bats are aerial insectivore. The noctilionoids, including the fishing bat, which is Noctilia here, is a gleaner. And what it means to be a gleaner is that it actually is looking, I'm using looking, but it's actually echolocating over the surface of the water. And when it finds a signal that there's a fish, then it goes in and catches the fish off the surface of the water, this gleaning. And this gleaning is really fascinating because the noctilionoids, the neotropical noctilionoids, for the most part, most of the species that we're going to see are actually gleaners. So those that are insectivores, the majority of them are actually these leaf-nosed bats that actually catch these insects off the surface of a plant, off the surface of a leaf. So they actually sense it and get it. So this gleaning turns out to be interesting because, of course, it turns out that gleaning insectivory is what evolves the ancestor of the leaf-nosed bats, the Phylogenetic, and the ancestor of the neotropical noctilionoids. Here's a collection of different genes whose in model organisms, the function, or in cell lines, the function has been ascertained in the eye. Just to give an example, what are these two nodes, the ancestor node and the Phylogenetic node? What are some examples of this positively selected genes? So in the ancestor of all the noctilionoids, we've got groups of genes that are associated with retinal homeostasis and responses to salt stress. That's really very suggestive because, of course, if we think about eyes, blood pressure in the eyes, the homeostasis within the eye is actually quite a delicate thing, a sensitive thing, right? And it varies depending on the size of the eyes. And so we've got genes that are under positive selection there. In the Phylogenetic, we've got some genes that are associated with the regulation of gastrulation and body morphogenesis that were found to be expressed there. And there's more genes that are on other nodes, but you can see there's a lot fewer, right? When we get to the evolution of nectar feeding or the evolution of plant feeding, we find much, much fewer genes mapped to the eye in a model organism. So instead, what we find, we have to turn our thinking upside down. Instead of thinking about this evolution of these new ways of feeding, of this new foodstuffs that the bats were eating, we needed to be thinking about foraging. The kind of foraging that is actually very different, right? The kind of foraging that the leaf-nosed bats have, this gleaning foraging that they do, and as well as the fishing bat, which also has this gleaning evolution. So what we find is that this vision genes, this positive selection in the vision genes, is actually tracking the foraging strategy. So to summarize all of this, we started out thinking that adaptation was going to be tied with the feeding ecology, but instead what we found was the adaptation was tied with the foraging ecology. And this molecular adaptation is traced back to the evolution of a way of gleaning, of a way of hunting that's gleaning, and not as much to the specific diets. There are some that are tied to the specific diets. And you can see here, this is one of those specialists. This is a bat from the Caribbean, and it's specialized in visiting flowers. You can see it's completely covered in flowering. Oh, by the way, this week is Bat Week. It's like International Bat Week. So you picked the right week for us to hear about this. So I'm going to move on to the next one, and that's the one where we have like that little asterisk in the molecular, because it's not really molecular. I mean, we kind of included different, the phenotypes as well. So is it the case that the bat had imposed trade-offs, right? So of course, one of the arguments that we made when we were proposing to do this work is that if a human or a species, rather, a mammal species that is on the ground, right? If you're on the ground, you can conceivably, like, your head can expand. And we see radiations of different mammals, for instance, where the different species have different sizes, different relative sizes of head. But we thought, well, okay, in bats, you hit a hard ceiling pretty quickly, right? And you hit a hard ceiling because bats, not only do they have to, keep proportion with the rest of the body, but they also have to be light enough. And so there's a hard ceiling about how large a bat can be. And so far we were only talking about genetics, but we thought, well, the morphology, the logic of this was that morphology was going to be constrained. And do these constraints in technology translate into some trade-offs, that you cannot have a bat that has giant cochleas and big eyes and a giant olfactory bulb all at the same time. You have to make space. The head can only get so large. And do the timing of these inferred changes, does the timing actually correspond to the model that we just found with genetics? So what's one way to find out? And until rather recently, this was not something that could be easily done. So this is Ronnie Hall, who's a student in University of California, Merced, and he led this part of the work. And he took advantage of a method that I'm sure lots of people in the audience have used called DICE-CT scanning, right? And it's this scanning, the CT scanning, where you can actually visualize the soft tissue inside specimens, right? And my former student, Laurel Yo, who's now faculty at UNC, she actually pioneered a lot of these methods with bats, and we published a number of papers using these techniques, and she's published a number of papers using these techniques. And so we already had a backlog, we already had a database of this DICE-CT scanning that was comprehensive for this. And one of the cool things about this is that the journal where this came out, Evolution, actually did a little highlight and produced this really nice, did a little highlight about the work, right? Where we're testing this hypothesis that we had put forward about the quantification of these different parts. So in bats, the eyes are not embedded in an orbit like a primate like us. So as we can very easily see in the skull, when we just have the skull, you can kind of measure the orbit for primates. For bats, those eyes are actually kind of floating in the soft tissue. So we absolutely needed that space of DICE-CT. We needed this technique of DICE-CT to be on. And then inside, you have the olfactory bulb, which of course is associated with olfaction, or the degree to which the animals are using olfaction. And then of course, what bats are famous for is echolocation, right? And echolocation requires that you have these cochleas that are enlarged. So this sensory structure, the idea was, can we look for this pattern of covariation, and especially negative covariation that would indicate that at the comparative level, there would be some evidence for a trade-off, right? So we were thinking, logically, you can't just have all these things be gigantic size. And so I'm going to walk you through the results such as they are. So here's the interesting result. The most interesting result, so these have been corrected for the size of the heads because different species of bats have different sizes of heads. And so after correcting for that, we actually find a significant and positive relationship between the eyes and the olfactory bulb. So the opposite of what we were saying, no trade-off. Instead, the bat species that have larger olfactory bulbs also have larger eyes on average. But when we look at this covariation between the cochleas and the orbits, or the cochleas and the olfactory, we find no covariation, there's no signal. The cochleas are doing their own thing. They are evolving in their own merry way. That's completely different. So we asked the question, well, okay, so how do we get to fit everything in the head? So in relation to ecology, these are the proportions. These are the proportions that are found in the for different groups, different ecological groups. So the insect group is the outgroups in this case. And you can see that these outgroups just have really large cochleas, right? A lot of the real estate in this morphology is taken up by the cochleas, and then the olfactory bulb, and then the eyes, right? But when you get to the neotropical leaf-nosed bats, which are the ones that are shown, the four on the right, then we see that even the carnivorous bats have these carnivorous bats have like olfaction taking over. More than half of this volume is being taken over by olfaction, and the cochleas are smaller. And the cochleas get smaller and smaller in other groups. They're even smaller than in the carnivorous bats, in the nectar-feeding bats, in the omnivores, and in the fig-eating. And in the fig-eating bats, we find the largest eyes of all. Got this super large eyes. So thinking, well, it all fits in the head because these bats are switching from a majority cochlea. The descendants of the ancestor Phylostomid are actually switching from the cochlea taking up a lot of real estate to the eyes and the olfactory bulb taking up a lot of real estate, even though we don't find that. So the next step, was to ask, well, when did this evolve? We've got, once again, color-coded those different nodes that correspond to the phylogeny of the group as a whole. And we see that off the bat, the Phylostomids, the ancestral Phylostomid is inferred to have a larger olfactory bulb, as it's highlighted. And it's also inferred to have smaller cochlea, but that difference is not significant, right? You can see that the cochlea is smaller, but the median is kind of overlapping very much with the ancestor of the entire clade and other families. You can see B, that the ancestor of the Phylostomid also has larger orbits. So already this ancestor can be inferred to have larger olfactory bulbs, larger orbits. And one curious thing that we weren't expecting at all, because remember, we didn't really find a very clear indication of how the cochlea were co-varying with other parts of the morphology, is that the nectar-feeding bats have the smallest cochlea of all. They just evolved to have this really tiny cochlea relative to them. So in this part of the work, what we saw is that we expected these very hard constraints, and these hard constraints would lead to an association in shifts related to diet, right? Like we might find different negative correlations, and these negative correlations would change depending on the diet. But instead, what we find, we find that there's a positive correlation instead of a negative correlation between the olfactory bulb and the eyes, right? And the cochlea seems to be evolving separately, except that in nectar feeders, the cochlea seems to be minimized, right? There seems to be an omnivorous sensory morphotype. What do we call an omnivorous? This ancestral neotropical leaf-nosed bat seems to already have big eyes and already have a large olfactory bulb, both of which on average seem to be associated with including plants in diet, right? So in the first paper that I showed you that was led by Kalina Davis, we talked about evidence of pre-adaptation. Is it possible, right, that this ancestor had evolved this for some other reason that we don't quite understand, had evolved this positive selection at certain genes, and was thus pre-adapted to later on take on this new diet. But in this paper, we were thinking, well, what if instead, and this is a hypothesis that had been proposed first in the year 2000, what if instead there is a foraging shift in the ancestor, and this dietary flexibility, this ability for the bats, has the bats, instead of catching the insects in the air, but catching the insects off of leaves, when they do that, they start coming into contact with plant material and plant material and plant material, and then selection takes over, right? And so there is this dietary flexibility that is associated with this foraging shift from the beginning. And so it's no longer pre-adaptation because it really is associated with the foraging shift, right? So this is also very interesting because in a separate set of analysis, we were looking at what was at the time the oldest fossil to have been known, or to have been inferred rather, the oldest fossil to have been inferred to be associated from a bat that's associated with a plant. And that oldest fossil is shown in the top right. It's called Palinophyllum, and it is from the central valley of the Magdalena River in Colombia. And in that valley, this species is known from only 2 teeth, that's all. However, the teeth were diagnostic, and the proportions of the teeth are such that when it was first described, it was immediately described as a nectar feeding bat, right? Immediately you can place it, it's a 12 million year old nectar feeding bat that we have in our hands. So a bunch of years ago, I wanted to kind of try and find out more about this bat, right? Is it a nectar feeder? Is it a generalized nectar feeder that includes other types of foods in its diet, especially insects? Or is it a more specialized nectar feeders? And we used phylogenetic regressions to actually infer this dietary association, finding that the proportions of the tooth are such that this too is an omnivorous bat. This too is a bat that includes more than one thing, insects, and it also including plant. And we had this artist, Michael Hansen, and everything that you see in this picture, every part, the Hoatzin, every species that you see here has been found in the fossil record of La Venta in the Magdalena Valley of Colombia. So he painted this picture of this bat getting nectar from the flower and also getting insects. So this tells us that even this ancient, of course, this is not the ancestor of all of this, we also did a bunch of work to put it in the phylogeny, and the phylogeny tells us that it is not the ancestor, it's closely related to a set of nectar feeding bats from South America. But it gives us a picture of this flexibility happening pretty early on in the history of this group. All right, so that takes me to the last bit of this, and it's interesting because, of course, again, the sense of smell, the perception of smell has now become something that lots of people are interested in because it can be diagnostic of a series of things, right, including viral diseases, but also for other reasons. The perception, the sense of smell is one of those things that starts degrading way before there's other neurodegenerative diseases. People start perceiving smells differently. So it's indicative of things that are going on in the brain way sooner. So there's a bunch of interest there. The question that we're asking is simpler here. Is just, does the diversity in these receptors, these olfactory receptors, is it related to the diversity of diet? That's a question that's surprisingly hard to answer. So let's walk through a little bit like how these olfactory receptors work, right? So mammalian olfactory receptors are very diverse and they're fascinating, right? So this is a model of the receptor embodying in a biological membrane. So inside of our noses, we've got these neurons that express olfactory receptors, and they just express one olfactory receptor. It's one of the few tissues, neurological tissues in your body that as an adult, it continues reproducing, right? And so you have a continuous coming in and updating, right? A continuous reproduction of these neurons that are expressing that one olfactory receptor. So far so good. It's just an olfactory receptor on the membrane of this neuron inside the back of your nose or inside your nose. And after binding, the receptor activates a switch that communicates, that starts the mental representation of the odor and communicates with the olfactory bulb, which you saw is kind of like right in the middle, right, of your skull, or rather in our case, it's kind of right behind, right? So you have these volatile compounds that float in the air. The volatile compound binds in a kind of lock and key type of fashion, or that's the best hypothesis that people have right now. And once it binds, that representation happens. So that literally the sense when we are smelling something, we are actually binding to it, right? There's more complexities to it because, of course, you can have different volatiles binding to different places and creating like a multi-receptor perception of smell. But this is what's necessary. This is takes us to the point that I'm trying to make, and that's that these receptors, the diversity of olfactory receptors in the genome, tends to reflect the diversity of the odorants. So, for example, here's the mouse genome. You notice a couple things. So, first of all, the olfactory receptors, as far as protein coding genes go in mammals, they take up between 3 and 5% of all the protein coding genes. They're the single largest family of protein coding genes that you have in your genome, including in us humans, even though in us humans, a whole bunch of them are pseudogenized. So we see them here. They are clusters. There are clusters in the genome of the mouse. There are clusters of olfactory receptors. They are in groups, in tandem repeats, and each receptor protein is encoded by one olfactory receptor gene, right? So if we hypothesize that the mouse perceives, for example, another rodent, but it also perceives volatiles that are put out by cats, volatiles that are put out by fish, volatiles that are put out by waste like hamburgers, right? By this waste, and there are receptors that are going to be binding to those odorants. In order to get that mental representation of what the mouse is smelling, there's going to be receptors that bind to those odorants like a lock and key type. Sometimes not quite as tight, but you get the idea, right? And so there's loads and loads of them, and they're in these arrays. There's a tandem array, so they repeat one next to each other. And of course, if you're studying genetics, you imagine what happens next. And what happens next is that if genes are next to each other and they're really similar to one another, one of the things that happens is mistakes are made. And this mistakes means that these genes often duplicate in total. The entire gene just gets duplicated. And at the other end, these genes are often lost in total as well. But this process of gene duplication and gene loss is actually very hard to study. The data collection is hard. So we've got this example from a few years ago, and in this example, what they were trying to test was this idea from primatology that will be familiar to some of you, that primates such as ourselves have incurred some kind of sensory trade-off in which we're more visual and less smell-driven, right? And therefore, there's going to be a lot fewer functional olfactory receptors, and there's going to be this trichromatic vision that enables most humans to actually view a particular range of color, right? And so this is a test of that. And you can see that the number of species that they have is relatively low. They have relatively few species. Part of the reason is that they needed very high quality, highly contiguous genomes, because you've got this mistake. So you can't easily, if you do a low coverage genome, you're going to hit some olfactory receptor, but do you know where it belongs? And do you know if it's a paralogous copy of another? You don't. You actually need this high contiguity. And that makes things very expensive, right? Because if you have a highly contiguous genome, it's going to cost you anywhere on the vicinity of like $30,000 back then. Nowadays, probably maybe $20,000 will do. The analyses are actually really hard. Within a still species, super widely used models are still not fully settled because, as you can see, olfactory receptors are just hyper diverse, right? There's hundreds. Sometimes I talk with people who study olfactory receptors in insects, and they say, oh, this hyperabundance, I've got 40. And I think, well, in mammals, right, bats have very few olfactory receptors, and we've got 300. So we've got order of magnitude differences on the whole. So one of the problems, so to bring that price down so that we could study the olfactory receptors, but we didn't need to shell out between $20,000 and $30,000 each time we wanted to study a different species or even a different individual, we decided to come up with a solution, right? So here's a very high quality genome at the bottom of this graph. It's a very high quality genome of the vampire bat that at the time when we started this part of the work was one of the best genomes for bats that was available. And then you have with Sanger sequencing, and this is going to date me, and is that with Sanger sequencing, you can actually do PCR of the olfactory receptors and then clone and then sequence them with one of this old-fashioned, right? And you can see how those primers work. They're actually severely biased, right? Instead of covering the different olfactory receptor subfamilies, which are noted here at the bottom, they cover only a few of the subfamilies. So they're taking in a very, very biased part of the diversity. We could also just go and take bats from the field, which is not my favorite thing to do, but has become indispensable for some of these questions. And we could take transcriptomes, right? We could take the noses and take the epithelium and get the transcriptome of what is being expressed at the moment that this bat is going into, that this bat is being collected, right? And so we got these transcriptomes, but of course that's going to be biased in a different way because we don't really know what's happening, right? Like it's going to vary from one individual to the next, what is being expressed and what's being conserved. So what we came up with was an affordable solution called sequence capture, where we are binding and pulling down the genes. And we were able to, despite the different pieces of the genes being similar to each other, design an affordable solution that brought the price down from $20,000 to $200 for each individual, right? So it's like a huge saving, and we get about 90% of the olfactory receptors in a pretty much unbiased manner, or unbiased compared to the other methods. So once we had this toolkit in our hand, that was led by Laura Yo, who's former graduate student. And so once we had created this approach, we could actually go back to these comparative samples and even to population genetic samples, but I'm going to show just one comparative example here, right? And we've got these 3 sympatric species that together live in Costa Rica. The one at the bottom is the largest one. It's called Carolia perspicillata. And it's got about half of its diet is pepper species, and the other half is just other foodstuffs, including some insects. We've got at the very top, the specialist, Carolia castanea, and 95% of its diet is just species of the piper plant. And then there's an intermediate that falls in between the two, and they all live together in the same forest. You can see them flying side by side. So we were able to get this repertoire of olfactory receptors very cheaply from each of these species and from actually many individuals of these species, but I'm just showing part of this. And you can see that we get this olfactory receptor subfamilies across all of them. So we used a phylogenetic approach to try to investigate if we have, which one ends up having greater diversity. So first, we get these ortho groups that are, like phylogenetically ascertained which ones are closer related. It can show us that for the specialist, which is castanea, which is shown it's very dark green. For the specialist, we've got lots of paralogous copies of the same gene, right? Lots and lots of paralogous copies, right? So you end up having, if you use a Shannon-Wiener index, which is borrowed from ecology or borrowed from information theory, and we use the Shannon-Wiener to quantify what the diversity is, you end up having lower diversity in the specialist because the specialist has lots of the same thing, right? But for the generalist, Carolia perspicillata, we end up having significantly more diversity measured by the Shannon-Wiener index. Instead of finding lots of copies of the same thing, we find a few copies of lots and lots of these different groups. So in short, we were able to demonstrate that this diversity of diet that has been investigated for these groups is associated, at least for these bats, is associated with a different diversity in the olfactory receptor genes. So molecular diversity in the genes is related to the diet diversity. Do we know about positive selection and adaptation? We do not, because that requires an extra layer of modeling that is not very good, right? Like once you're throwing paralogous genes into the mix and you can't really tell which one is the paralog and which one is the ortholog, once you're looking at that, it makes it really hard to look for molecular adaptation. So this process of duplications and losses make it difficult for us to ascertain that, but that's part of the work that we're doing, and especially with this toolkit that we've developed, we've been able to also study this diversity within species as well. Here's a picture of Carolia castanea. This is not a pepper plant, but this is how they eat. They actually eat like corn on the cob. So what you see is one of the fingers with a claw is shown holding the food. All right. So to wrap up, does feeding ecology shape evolution? Does the bat have imposed trade-offs? Does diversity correlate to diversity? Well, feeding ecology doesn't shape the evolution of these vision-related genes, but instead foraging. Foraging is what's really shaping the evolution of these vision-related genes. We do not find evidence for trade-offs or negative correlations. We instead find evidence of positive correlation, whereby the olfactory bulb and the eyes are correlated, are evolving together to become larger in this species of neotropical leaf-nosed bats. Diversity does beget more diversity. So more diversity in diets corresponds to more diversity in olfactory receptors. Do we know if it's adaptive? We do not know if it's adaptive, but that's our next frontier. And with that, I would like to thank you for being a fantastic audience, and of course, the members of my lab who make all this work possible. Thank you.
Thank you, Liliana. Well, this is really very interesting, especially very compelling type of work that you guys are doing. So with this, I will just open up for questions from the audience. If you have a question, please, you can unmute yourself if you want, or you can use the chat. We already have some thank yous from the chat. So Sean Pierce, do you want to ask your question or you want me to read it? Okay, if I don't hear anything, I will read this question. To Sean Richards, it says, regarding the bags that are fishing, how are they able to target the fish with echolocation if the water surface is relatively flat? And are they targeting ripples made from fish movement?
That's right. They are targeting the ripples that are made and can perceive. Remember that these bats are, emitting the sound, right, at very high volume, right, and at very high frequency. And so we're perceiving that. There is work from a lab, from Yossi Yuval's lab. I think they got a Human Frontiers of Science precisely to study the water like a mirror, right? What happens when the water is like a mirror. But yeah, the people who study the behavior of the fishing bat have demonstrated that they are echolocating for the ripples. Yeah.
Okay, thank you. I don't see any other questions. Everything says very cool. Matt Holding says, what was your strategy for evaluating signals of positive selection in these large gene families when the content birth rate confound determination of homology?
Yeah, that's exactly the problem. Thank you for defining the problem really well. This is a very good way to define the problem. So we are working, this is something that's very preliminary right now. So we are working on within populations, right? Like we've got a sample of like lots and lots of individuals of those three species, right? I shared your pain, and that's why I asked, oh, okay, we can have this pain forever. We can have this pain together because it is very complicated. So we came up with a couple of different strategies, one that we call like a bag of genes, right? And so we basically subdivided the genes, making calls as to whether, a subfamily, and this is, when I say subfamily, I mean a gene subfamily. A gene subfamily is a unit that we can study, or a gene subunit within an individual or something. We partition the variants in different ways to try to ascertain that. But I would love to work with someone with much better population genetics chops that would like to go down in this direction. Because one of the things that, and I think the person who's asking the question is that these models, the models simply aren't there. Like the reason why we're doing this kind of seat of the pants type of trying to put them into different bags is that even if we knew with great precision, which we never could actually, because of the duplicative nature of the genes, if we knew with great precision who's the ortholog of home, right? The duplication process itself is not very well modeled. It's not the population genetics of that. So there's an opportunity. We have the data, and we have made some headway into ways of thinking about this. But this is something that I'll admit, I am not a population genomicist, right? My forte is the comparative realm. And so that's why I'm showing comparative results. That's why the comparative results are published and the population genetics results are not published yet.
Excellent.
I love the sharing of the pain.
Yeah, so if one was to mirror a similar study of a structure to prey choice using the DICT scanning you use, what suggestions would you have for making sure you adequately cover all aspects of prey capture to not leave something out?
Oh my God, this is such a cool question. So we got a paper that's in revision right now, and that's kind of like the next stage of this, where we were looking at modules of the head, right? And we were separating into sensory modules, which is relatively novel. There's a lot fewer publications having to do with sensory modules, and biomechanical modules. And these biomechanical modules are something that lots of labs, especially at the comparative level, have been doing for a long time because, and like for example, in fish, like elongated skulls and less elongated skulls, and we see the same in bats, right? This elongation, this kind of, and we see it in carnivorans, et cetera. Like wherever you look, there are studies that take the hard CT scans, and you can correlate that to their diet, and you have this biomechanical component, right? And so we're actually looking, okay, so the paper, so if you're in the audience, please help us get it accepted. So because, so we've just resubmitted that paper, and that paper is fascinating because it tries to get at this, like at capturing both the foraging and the biomechanics, right? And trying to ask the question about where the correlations are and where the correlations break down, right? Because of course the modularity means that some parts of the specific morphology are more correlated with each other than others. And there are different schools of thought. This is like I'm completely speaking about things that I'm not super confident on, but there are different schools of thought as to whether having the correlations is actually going to limit the diversity that we see, right? The diversity of the morphotype, or having the correlation could actually contribute to the diversity. And so stay tuned, because we are using precisely the DICT to capture all the aspects that we think are associated with this finding the food and then processing it.
Amazing, thank you so much.
Fishes, yes. Yeah, fishes, yeah, exactly. So fishes are really, yeah, of course, much more diverse, of course, bats. It's only so many bat species, but I guess from where I'm sitting, infinite number of fishes. Inordinate fondness for fishes, I guess, yeah.
Yeah, I don't know if anybody has any further questions right now. If not, I just couldn't wrap around. Maybe I just couldn't understand why, but I couldn't wrap around why olfaction and the size of eyes. I mean, why that correlation? I mean, why
do we have that?
Yeah, what do you have that in this type of bats in particular?
So for decades, ecologists, like pollination ecologists and fruit dispersal ecologists have characterized certain plants as being bat plants, and certain plants as being like the ones that are associated with a vertebrate, right? Some being bird plants, some being, so in terms of fruit, a bird plant is going to have this very bright color, right? When it's ripe, it's going to be advertising, it's going to be doing the things. How do you do that in the dark? Right, which is when the vast majority of bats are active. You do that with smell. You do that with the sense of smell. And then in terms of pollination of the flower association, there's also bat flowers and there are bird flowers, right? And the bird flowers like open by day and they have pretty colors and they do this. And then the bat flowers smell like a corpse when they're ready to be pollinated, right? So we think, so the part that I'm most intrigued, so basically we can, obviously there's a lot more testing to do. There's a lot more work. There's just, this is kind of like scratching the surface. There's a lot more that we can study, like within populations and looking at, there is one species Anthrozophis pallidus, the pallid bat, that's going down a very interesting road. It's like in Arizona and in Texas, and in the southernmost part of its range, it's decided somehow to start eating nectar from flowers and fruit. And so it's really fascinating because, of course, these bats are like super specialized, millions of years, whatever. And so they're like super elegant, like they get to the flower and they have a hovering flight and their head goes in. And this bat, I encourage you to look at, okay, so I'm going to type the name. So pallid bat. If you look online, there are YouTube videos of this pallid bat, like clumsily, like it's a bat that like eats scorpions, right? And it's kind of landing on the cactus, really kind of like, and you know, the idea is that maybe the nerve receptors aren't as abundant. It's like landing right on the thorns, right? And it's kind of a mess, and it's kind of like putting its head and just eating everything. And it's just not really adapted to do that, or it doesn't look like it, but it's still switching that diet, right? It's still going in that direction. So anyway, so I would be fascinated to see in this within population context, if we start seeing some of this difference. And of course, the olfactory receptors evolve really quickly, right? Because one of the things about these duplicative genes is that they evolve a lot more quickly than the rest of the genome, right? So anyway, that's kind of far-fetched because I don't know if, you know, we don't know how many generations these have been going on for. But yeah, so there are bat fruits and there are bat flowers and there are others that are clearly not targeting the bats. And the ones that target the bats use the sense of smell to broadcast, you know, like, so the bats that have been studied really carefully for behavioral analysis, the bats are figuring out where the right fruit is generally with the scent. And then as they approach the fruit, they echolocate to find exactly where the fruit is. Yeah.
I think we have one more question. It says, do you
have any reason to think, oh, that's a really good question.
That the cost of flying with giant ears or special noses is important
ears.
to bat?
They change the shape of their ears. They change. I would say that there are people like Charlene Santana's lab, who's at UW. I think she was trying to look at, she was trying to get into like the nose sleeve, right? Because you can also see this variation in the nose sleeve. And like what is exactly what the nose sleeve is doing is also kind of a matter of debate right now. But anyway, she was trying to do that. And one of the problems is that you have a specimen and the specimen, right, looks a certain way. But when they're actually flying, they change. They literally, there are carnivorous bats, for instance, that actually, their nose sleeve, they actually kind of pointing downwards, right? And so the shape that you can get from a specimen and the actual thing that they do when they're flying are two different things, which doesn't make it impossible because, you know, people have these tunnels, right? Like you can kind of put monitors and sort of track them. You're not going to get it for lots and lots of species, but for some species, you might be able to use this very good setup, so fly it, right? To kind of see how they are reshaping those ears in flight. It's a great question. I'm sure somebody has done a little bit of that.
Okay, well, thank you so much, Liliana. It's been a great talk. We all clearly enjoy it, and I hope you also enjoy the questions. And we can wait to see you here sometime in the future.
Awesome. Yeah, likewise. I thank you for inviting me and enjoy Bat Week.
Okay.
Okay, bye.
Bye-bye.